The full list of talks for the quarter is up on our website at def.ucla.edu. And I have a couple of announcements. Next week's talk is by Michael Alvard, who's at Texas A&M, and he's going to be talking about social structure, cultural kinship, and cooperation among the Lamalera whale hunters of Indonesia. So be sure to come and join us for that. And then there's one additional talk that has been added to the schedule that is not going to be the regular back time. It's going to be on Wednesday, December 8th, which is actually during finals week. We hope that you'll all come and join us because it's Christoph Bosch who's going to be coming, so that should be a really exciting talk. And the title and abstract of that will be up on the website shortly. So this week we are happy to welcome Jamie Jones from the Stanford University Department of Anthropology, and he's going to be talking about contact networks and models of infectious disease.
Thanks, Brooke. Thank you. Some of you may have noticed that I did a late substitution. I was going to talk about foraging theory, and I have some results. I just don't have a very polished talk, and I just happened to have a talk on this that I thought also may be of interest, probably more of interest to some, less of interest to others. Before I launch into it, I always do my acknowledgments at the front. That way I know I do them. And everything I'm going to talk about today has been done jointly with Marcel Slatte, who was a Society and Science Fellow at Stanford for the past 2 years and is now an assistant professor at Penn State in biology. A bunch of the work that I'm going to talk about is done jointly with some colleagues in computer science, particularly Phil Levis, who's in the CS department at Stanford, and in biology, Mark Feldman, and everything is jointly with Marcel. There aren't many intellectual breakpoints in one's career where you can absolutely identify that's the moment where it happened. And for me, one of these is where I first got interested in social networks, and it was in 1995, and I was a TF for Richard Wrangham's primate social behavior class, sitting in the basement lecture hall of William James Hall, and we were actually talking about a paper that Gene Altman and Susan Alberts had written on dominance hierarchies, and for whatever reason, this is what I don't remember, I had just been introduced to graph theory. And I was sitting in the back and, sort of spacing out a little bit, and I was looking at these pictures of baboons doing their baboon things and thinking to myself, geez, I bet we could represent their dominance hierarchies as graphs, and that might even be useful. And I pursued this for a little bit and then sort of left it behind. And when I was looking to do a postdoc, I ended up, very fortunately, to stumble into a really amazing group at the University of Washington where I learned quite a bit about social networks and infectious disease and statistics. And I'm going to talk to you about some work that we've undertaken at Stanford to try to use social network models to understand disease transmission processes. But I know that people here are interested in cultural transmission, and the substrates of social structure that diseases diffuse across are the same ones that ideas and political influence diffuse across, and so I think that there's a lot of potential here for thinking about cultural evolutionary questions. As well as other evolutionary questions as well. So the basic model that we employ in this particular area of work that we do in my group is we're ultimately primatologists at heart, and we do this sort of ethological observation of some sort or another. And this may actually be good old pen and paper and binoculars observation, or it may be what we might call technologically enhanced ethological observation. But there's an element of actual observing behavior on a relatively small scale in a fairly fine-grained sampling environment. We construct models of social structure based on our observations, and these models tend to be graph theoretic. They're not always, but frequently are graph theoretic networks. We do stuff on top of these networks, right? we diffuse things and look at processes that happen on top of networks. This is it doesn't really matter, but this is a simple model for the transmission of 2 different retroviruses among red colobus monkey populations in Uganda. And then we look at outcomes. Right? And frequently, in the infectious disease context, we're looking at things like final size of the epidemic, the number of individuals who are infected when the epidemic has run its course. And that's what this is here, is an epidemic curve. So we use mathematical models and computational models to look at processes that happen on these social structures. And what do models do for an anthropologist? Models, most importantly, let us experiment, and they let us experiment in ways that we couldn't do for ethical reasons and for practical and logistical reasons. If we wanted to, if you're looking at infectious disease, it isn't like you can, go in and infect a population, particularly of people, and see what happens. Despite, crazy paranoid theories, it wouldn't happen. At least in our world, it wouldn't. And so mathematical models give us a way to experiment. Here's the result of a very simple, susceptible, infected, removed epidemic. What happens is that initially everyone is susceptible, and then the epidemic comes in, it sweeps through, and people are no longer susceptible because they get infected. And they move out of the susceptible category, and eventually they fall below this line here, this dotted line, which happens to be the epidemic threshold in this case. And then eventually the epidemic dies out, and we get a sort of equilibrium here. Right? And so this particular SIR model is a very simple one. It has no demography. There are no births or deaths. The population can't renew itself. And what we see is that we just get this very steady equilibrium. And we can ask a question like, what happens if we add population renewal? And we get something like this, right? We see that, it drops down just like it did before. But then eventually the population replaces itself. So you could think of this as being something like a childhood epidemic, right? Something like measles or mumps or rubella. And eventually, when the population crosses the epidemic threshold again, we get another little outbreak, and it'll decay back again. So this is a very dumbed down sort of example of the type of experimentation that mathematical models allow us to do. We can look at more exotic behavior. Jamie will recognize this maybe as this crazy plot that Anderson and May put together to demonstrate some of the exotic behavior that simple epidemic models have, which I've reproduced in a prettier color than in their original. But that's basically the idea. So that's sort of the third step in our, so I'm going to work backwards a little bit. The third step in our sort of formula for investigating these types of phenomena. The standard model for an epidemic is really remarkably similar to a model for chemical kinetics, right? As absurd as it may sound, you can imagine you've got this supreme being, perhaps, who's got a giant Erlenmeyer flask, and into this flask he or she can pour in susceptible people and infectious people and swirl it around, and that swirling, that's our social interaction, right? Because you're causing those molecules to move around and come in contact with each other. And then you count up how many infections this produces. It seems like an absurd model for some sort of a human process. It turns out it works sometimes. For things like diseases that are transmitted by aerosol or by droplet, through casual contact on subways and public transportation and in crowded football stadiums, this is actually not a terrible type of model. For other types of interaction, it's not as good a model, right? So in the case of a droplet transmitted thing, right, we find that the number of new cases is frequently proportional to the number of people who have the infection. Whereas in something like a sexually transmitted disease, the number of new, say, gonorrhea cases is not proportional to the number of people who have gonorrhea, because it requires a very special type of contact, not a casual contact, to transmit this particular sexually transmitted infection. And so this, I think that this is really where anthropologists and other social scientists committed to looking at actual behavior can really make an impact on social network type models, is by thinking hard about the relations that define our networks. This is something that I think is far too often taken for granted, and the difference between a disease that's transmitted by droplet and a disease that's transmitted by intimate contact is a really, I think, profound way of thinking about this. So we have these processes that happen. We have relationships that we can define, that will define our social structure. And here I've just plotted a graph, right? So the red dots here represent individuals. They're the nodes of the graph. They could represent some other type of entity, like an organization, but in this case, we're thinking of this as individuals. The edges that connect nodes are some sort of social relation, some well-defined social relation. In social network analysis, frequently, probably the most common type of relationship is friends with relationship. Right? You administer a social survey and you ask, are you friends with, and maybe you have a list of names in a school or something, right? So you have some well-defined social relationship. And in this case, we see that there are a number of individuals who have lots of contacts, right? They have lots of friends or lots of sex partners, or they sneeze on lots of people, whatever our social relation is. But most people have very few. And this is a model that's gotten a lot of mileage in the last decade or so. It's a power law model of the degree distribution, and the degree is simply just if you count up the number of edges that come out that are incident to any given node, that's simply the degree, the number of contacts you have. The degree distribution follows a power law distribution, meaning that most people have very few contacts, but a few people have a lot, and they form these sort of hubs. And if this is a sexually transmitted disease network, you don't want to be in this giant component in general, right? Even if you are someone like this person, probably a woman, who only has one contact, right? You've got some bridge here into this very high-risk component that we frequently see in these type of high-risk networks. So my metaphor for this is one way of representing this type of social structure. Something that you'll frequently see, though, is a marginal representation of this. And my metaphor for this, I don't know if it's helpful, but I imagine a giant squeegee, and we take our squeegee and we go, and we bring all the degrees over to the margins of the plot, and we make a histogram out of it. Now notice that we've lost most of the social structure by doing that. We can sort of compactly represent one part of it, right, what the distribution of the number of contacts is, but we've eliminated all the sort of the higher order, the transitive relationships, the cycles, and the things that are really kind of interesting to sociologists and anthropologists. And this is a marginal depiction of the same contact graph, and we can see that the great majority of people, over 80%, have a very small number of contacts between 1 and 2. There are a few people out here, we can't even see them unless we put this on log axes. There are a few people who have a lot of contacts, right? And those are the people who form these stars in these networks. You'll almost never see this representation in the literature. You'll never see a histogram unless you actually happen to look at Mark Hancock's and Mark, of course, just moved here to UCLA, lucky you. He's a real leader in the sort of intersection between statistics and social network analysis. And he's a really incredibly smart person who's taught me an awful lot. Unless you happen to look at Mark's and my 2004 paper, you'll never see this type of relationship plotted. What you'll see is something that looks more like this. And what this is It's a plot on double logarithmic axes of the complement cumulative distribution of your degree distribution. Okay, so it's the log survival function of that distribution. And what you have along here is the logarithm of the probability that you have, in this case, 1 or more contacts, 2 or more contacts, 3 or more contacts, and so on. When you have a linear relationship like this on double logarithmic axes, it's suggestive that you might have a power law distribution, right? Because you've got the probability is some power function. You take the logarithm of that. That makes it multiplicative and linear. And so frequently, what will happen is that people will estimate this power law exponent as the slope of the ordinary least squares regression line of this particular plot. There are a few problems with this. And the reason people do this is because these scaling exponents are interesting because they tell us with very little data, they tell us, with very little information about actual social relationships, they tell us a lot about the connectivity of graphs, for instance. The problem with this method of estimating is that the slope is a biased estimate. It's, the uncertainty is greatly underestimated. And when you look carefully, the data don't actually fit very well to these power law models. And this particular problem is what got me particularly interested once I had adopted social network analysis as something that I do, in thinking hard about what the relations are that define these graphs that we are interested in as the representation of social structure on which the processes that we actually care about happen. So I have a little rogues gallery of some different graphs here that depict quite different social relations, and you'll see that they look different. They have a different topology, okay? Or in a more sort of old-fashioned anthropological term, a structure, right? So anyone want to hazard a guess what this might be a network of? Straight spaghetti with blue meatballs? Something completely unilluminating. This is the California Institute of Technology. It's the Facebook graph, friendship graph from 2005, back when Facebook was a thing that was done within universities. And so there are 800 and something some odd students here, and they have an average number of friends of about 100. And when you have that many nodes and that many edges, you get straight spaghetti with blue meatballs. You get essentially no value to visualization of a graph. And what we'd see is if we plotted the marginal distribution of this, we'd actually have something that looked actually remarkably, I mean, you could call it normal. I mean, I'd say it looks a little more Poisson because these are counts, right? But it has a mean at about 100. No big tail. Right? No big tail. There aren't lots of stars here. There are a few people who have more friends than many, but there are very few isolates. Right? Here's one guy who only has one friend. Poor guy. And that's at Caltech. Ouch. No offense, Cameron. Okay, so here's a very different type of relation, and we see that people don't have as many of whatever this relation is. Anyone care to hazard a guess what this is?
Sex.
Sex? How'd you guess, Brooke? Right. So this is a sexual network. This is actually the largest connected component of a one-year sexual network in Malawi. One of the things that's really striking about this, we've taken away all the other components to this, is what we call these big dendritic formations, right? These very long branching structures, and this arises because people don't like to have sex with their partners. There's a social rule there, right? Maybe it's not a social rule, but it's sort of implicitly a social rule because you'll get in trouble. Like this poor guy's about to, from his brother who's about to off him, right? And this famous scene from Dante's Inferno. And so when you avoid making these short cycles, right, having sex with your partner's partners, then what happens is that your network sort of folds out on itself. Now, every once in a while, someone violates that rule. We don't know who it is, but you form these very long cycles. And this is a term that Jim Moody and Kate Stovall and Peter Behrman have termed chains of affection, looking at high school romantic relationships, where you see lots of very long dendritic cycles like this in American high schools with romantic relationships. So this is a very different type of social relation that gives us a very different type of social structure. Okay, so I'm going to talk about the structuring of social interactions, and I had a football game picture here of a mob. This is just taken from yesterday's paper. And this is a crowd shot from the Rally to Restore Sanity and/or Freedom in Washington, D.C. this weekend. And I actually think it's a much better model of what I'm trying to get at than a football crowd, because a football crowd actually has a lot of spatial structure because you've got a seat, right? Here we've got, a mob. We've got a group of people who are sort of amorphous, and they sort of mill about, and there are lots of them, right? Everyone has lots of contacts as they mill about, and there's no real structure to this. This isn't so much the way human social interactions work, right? The mob, the crowd, the undifferentiated horde, these are fairly rare events in the course of human daily life, as are these incredibly regular structures that we see in like a school play yard in China, right, where you have an extreme, these are kids doing calisthenics, and they're extremely regularly spaced. This is also a very rare type of social relation that we see, a rare type of spatial clustering of human interactions. I think what's really a very strong sort of metaphor for thinking about human spatial organization and how space structures human relationships is a cocktail party, right, where you have a couple people talking here and there, you have some bigger cliques, you have some smaller cliques, you have some isolates. I like to think of this guy as being me, sort of looking in and saying, who can I talk to? And you've got physical constraints on the size of conversational cliques, right? Because there's ambient noise that makes it difficult for too many people to get together and talk. There are frequently environmental or social constraints that shape our social interaction, and we have these sort of fluid structures that are quite clustered. Okay, and clustering is the key here. Clustering is the key element of social organization that I'm going to talk about for a little while here. So that's just a funky looking lattice of extremely regular interactions that gets folded on itself for no apparently good reason. This is my mob graph, right? Everyone has about the same number of interactions, and who they interact with is a random. It's just uniformly randomly chosen across the mob. And here is my cocktail party, which I've made an extreme cocktail party because it's amazing how quickly the human eye loses its ability to distinguish structure in these graphs. And so what I've done here is I've taken an extreme case where essentially we have these cohesive subgroups, right, where basically everybody is connected to everybody else, except for one person in each group who has one connection outside of each group. Okay, and this allows us to see the bunching and the clustering, and that who you're connected to is sort of a random function, and then there's a graph layout algorithm that plots things in space. Excuse me. And so you can really see the clustering, and I suggest that this is not as extreme as this. But this is really the sort of canonical type of human social organization that we have. You could think of these as conversational cliques at a cocktail party. You could think of these as households within a village. You could think of these as Because space is arbitrary here, right? It's just the layout is a function of the pattern of connections. There's nothing intrinsic in the spatial representation here. You could think of these as households within a town. You could think of these as towns across a landscape in a sort of larger level geographic space. Or conversational cliques within a party. Okay, so the question is, there's been a lot of work that's been done on looking at lots of different simple graphs, the type of power law network that I showed you first, or these random graphs. What happens to epidemics when we simulate them on top of these structures. But we haven't done as much work on looking at these more complexly, hierarchically organized, and I would suggest realistic types of social structures that humans produce. And so the key question as an anthropologist looking at these is, does social structure have a substantial impact on the outcome of epidemics? I think that most social scientists will say, well, of course it must. But it's remarkable how little work has actually been done on this. There are three very important domains of previous research on this, and the most obvious of this is individual heterogeneity. We know that heterogeneity in activity, the number of contacts that you have, can drive an epidemic all by itself, right? That variance contributes to epidemics. And this is a type of social structure. It's a very simple type of social structure, but it is a type of social structure. We know that assortativeness can have a big impact on epidemics. When you have strongly homophilic interactions, strongly associative interactions, this can really, for example, slow down an epidemic as it moves through a population, or it can slow down the transmission of information as it passes through a population. If you have disassortative interactions, you can actually, nothing's faster than random, but you can have more or less slowing of transmission as a function of assortativeness. And then there is a degree of clustering, where the clustering here isn't this hierarchical clustering that I'm talking about. It's really the propensity to close off triads. So if I'm friends with Brooke and Brooke is friends with Joan, then we close off this triad if I am also friends with Joan, right? But this is a fairly natural type of social interaction. And sociologists and social psychologists have suggested this is actually the sort of least costly thing to do, to close off these triangles, because it causes cognitive dissonance if you're trying to manage different sets of friends. And this type of clustering has been looked at by a number of people. But not so much this community structure. So most human populations are organized hierarchically, and by this I mean that most ties happen locally in a small group, a household, a village, and that these groups are embedded within larger communities, and that some ties happen with people outside the group. And we can think of this as being a multi-scalar process, households within villages, villages within regions, regions within nation states, and so forth. This varies by context, and some populations have far more structuring than others. There's a large and excellent literature on structuring in classical stochastic epidemic models, which is an area that in some ways I feel has been neglected by recent developments in network epidemiology. But that's all I'll say on that for the time being, at least. So our goal in this first thing that I'm going to talk to you about is understanding the impact of this type of community structure on epidemic outcome. So what do? We simulate lots of networks with different levels of community structure. We measure this community structure as Q, which is Girvan and Newman's measure of community structuring, which I'll show you momentarily. Here's where I think I think that if you ask Marcel and I what the major contribution here is, I think that we differ a little bit. And one of the things that I've been particularly interested in is controlling for heterogeneity. I mean, obviously we're both on board with this, but this is what I think is in many ways most interesting. We have regular graphs. We're simulating regular graphs here. Everybody has the same number of contacts. All that matters is the degree to which, or the probability with which a particular contact is within a cohesive subgroup or between cohesive subgroups. It's the only thing that we're varying. Okay? Because we know that contact heterogeneity has such a large impact on epidemic process. We simulate SIR epidemics. This is, as I mentioned before, susceptible, infectious, removed epidemics with these networks as a substrate. So 2 individuals can only transmit if there's an edge that connects them. Right? This isn't the giant Erlenmeyer flask model. We measure epidemic metrics, particularly the final size. In a graph context, this is the fraction of all the nodes that are infected at some point during the epidemic. And we evaluate the effective community structure. Our measure of community structure is this measure Q by Gervin and Newman, 2002. And the basic idea is that we've got, we sum across all nodes, we sum across all communities, so it's that EII is the fraction of nodes in community I connected to others in community I. And then we subtract away the squared value, which is essentially the fraction of edges in the network that connect to nodes in community I. Okay, so this is just a measure of the structuring when this number is very large. We have graphs that look like that first one that strongly structured one, where you have essentially cohesive subgroups that are connected by, just one or a very small number of between group connections. And this is what we get. These are 3 different levels of transmissibility, and I'm going to focus on the red one here. So we have extremely highly clustered networks. We have essentially here networks in which there's no connection between subgroups, right? And so what we get are only very small epidemics because there's no way for the pathogen to get out of that community and infect further communities. And what we see is that we have a fairly small epidemics for a while, and then we get this almost impulse-like response as we step down the modularity or the degree of clustering in our networks. And this was surprising to us because I'm unaware of anything that necessarily would have predicted this because, importantly, the basic reproduction number in all of these epidemic models, which is the thing that, it's the bifurcation parameter in a network, in an epidemic model, hasn't changed. Just the patterns of social connectivity.
Are these networks the same as like Duncan Watt's small world networks? Are they similar?
I mean, they're similar.
Okay.
Yeah. Mm-hmm.
And how are they different?
Well, so the algorithm that we use to construct these networks is we start off essentially with a Watt-Strogatz model, and then rewire until we hit essentially a target level of community structuring. Now, the thing that separates these 3 different, and so each one of these is based on 2,000 runs. And so the standard errors are actually on here. They're very, very narrow, right? And the thing with simulation studies is that you can just do more simulations and you can always bring your standard errors down. We tried to have a principled way of approaching this and just sort of a priori set 2,000 as a reasonable number. So the key here that gives us a real insight into what's happening is that as so these curves are basically what happens in models where the transmissibility is higher, okay? And so what's actually happening here is that we have, in this one, is that you have the infection gets into a cohesive subgroup. It bounces around, right, sort of at random within this group. And there's essentially a uniform probability that it will go out, it'll go along any particular edge in this group. Right? Now, most of these edges are within the group, and so infections are going to happen within the group. And it's very important to remember that this is an SIR model, so that recovery can happen. And once everybody recovers, then there are no more infections to seed further epidemic or further outbreaks in different communities. And so, with highly clustered networks, you get the stochastic fade out in the communities in which the epidemic is seeded. Whereas if you have a highly transmissible thing that doesn't fade out as fast, it can sort of bowl through quite a bit of community structure. You don't get the same sort of impulse-like effect, but you get qualitatively the same result. And here we've got a fairly complicated figure in which this is a typical outbreak in a non-clustered graph. They typically all kind of look like this, and it looks very much like an actual analytical epidemic curve, right? It's sort of more or less bell-shaped. It's a little asymmetric. It's got a little bit of a tail to the right. It's colored to represent the generation of transmission, right? So here the dark red is individuals who are infected by the index case, and then the next color, it's kind of hard to see under the projector, but the next color is the people who were infected by those who were infected by the index case, and so on. This one here, and we had to look for one, to basically rig a figure. We had to look because they were fairly rare, right? What happens when you do get an epidemic that persists in a highly clustered population? Well, it looks like this. Really, it looks like a string of more or less independent outbreaks. We've got lots of little epidemic curves here, and it just has to get lucky enough, from a pathogen's perspective, it has to get lucky enough to get passed from one community to the next, cause an outbreak, and just before it goes extinct, jump into the next community. Oh, sorry. And so we can see that here's a histogram of the final sizes for the non-clustered network, and this is a histogram, an extremely long right tail for the highly clustered network. Just to give you a sense of what's actually happening with the dynamics in each of these simulations. If you think back to this power law graph, I probably should have showed you another one to remind you of this. This is something that's always bothered me, is that we can say, well, it's heterogeneity in the number of contexts that's driving these big epidemics in power law networks. But the problem is that degree and betweenness, the centrality of individuals, are completely confounded in these sort of simple models. Individuals who have high degree in a power law network also are right in the center. They're stars, right? You have to kind of go through them in order to move from one part of the graph to the other. And it's bothered me for a long time. Is it really the degree that's driving the epidemics in these highly heterogeneous graphs, or is it actually the betweenness, right? The centrality of an individual who can act as a bridge for moving the epidemic from over here to down here and to up here and so forth. And what we see is that as our as modularity increases, the correlation between an individual's degree and their betweenness centrality declines, okay? So we have, and so here we have not a very structured network. Right, this is one without a whole lot of community structure, without a lot of modularity, and what we see is that there's a pretty good correlation between the betweenness and the degree of all the individuals in that graph. As we step up the degree of community structure, to something 0.876, which is right about on that steep part of the final size curve with respect to modularity, we see that this correlation really breaks down. And when we move up to the most community structured, we see that the betweenness and the degree, the correlation falls apart entirely. Because the people who have high betweenness, high centrality, this particular measure of centrality, are the people who bridge, right, this one community. Right? And they're not people who have a high degree, because of course everybody has the same degree in our simulation models. These of course are not, these are heterogeneous models. So in order to look at this particular correlation, we had to also do simulations that weren't on regular graphs. Okay. So there's that result. One of the goals of doing this is to try to develop ways of intervening in epidemics that are maybe more efficient, more effective than just going out and vaccinating as many people as we possibly can. This is particularly important at the outset of epidemics. And I'll talk a little bit more about betweenness before I jump into this next slide. We looked for what the best predictor of final size of the epidemic was, because we know who we're seeding each epidemic with, what individual we seed each epidemic with, and we can look at what correlate of the individual that's seeded best predicts the size of the epidemic, and it turns out to be this betweenness centrality. And so the question was, could we design an epidemic intervention that found high betweenness individuals? And we tossed around a bunch of different ideas for how we might go about doing this, and we wanted our algorithm to be something that would use only the information that's available during epidemiologic investigation. This is typically contact tracing information. Okay, and so the algorithm that we develop, we call the Community Bridge Finder, and I'm going to take you through how the Community Bridge Finder works in terms of how it performs, and then I'll tell you what exactly it is. And so the key point here is that wherever you have a red bar, it means that we stopped more infections using Community Bridge Finder than we did using this other method that's recently been proposed. There's a paper by Cohen et al. In 2002 or 2000, around then, and recently James Fowler and Nicholas Christakis have really been sort of pushing this as an effective use of social information. That basically it's just as simple. You ask people, you say, tell us a friend, tell us just any friend. And this relies on Feld's notion that your friends are friendlier than you are, and so if you ask someone to name a friend, your chances are they have a higher degree than the person than the ego does, right? And so if you vaccinate the acquaintance, then you get a bigger bang for your buck in a random vaccination strategy. And there are various elaborations on that, but that's the basic idea. And it does in fact do better than random vaccination. But our Community Bridge Finder actually beats acquaintance vaccination. And just a brief word about what these represent. These are back to Facebook again. We took the Facebook data from a number of different universities from 2005. We altered them a little bit because, the Facebook friend, what does that relation actually define, right? It's certainly not a real friend. It's your Facebook friend. God only knows who your Facebook friends are. But because we had other covariates associated with these network data, we were able to say, okay, so it's something somewhat more like an actual social contact if you're Facebook friends and you're in the same year of college and you're in the same major or you live in the same dorm. And so we pruned away all the relations that didn't fulfill one of those three criteria. And this actually anticipates the next thing that I want to talk about, which is the critical need for data collection, because we don't like using Facebook data, but what else are you going to do to look at actual human contact data for epidemic simulations? So these are different universities. that have different numbers of students, and the students have slightly different sort of Facebook behavior. And what we see is that wherever it's red, the community bridge finder stopped more infections than the acquaintance method. We see that for the most part, these are all red. It does very well. So what are the different vaccination strategies we could use? We could randomly vaccinate some fraction P of the population. We could vaccinate based on degree. We go out there, and since we all can identify immediately what our contact degree is, right? We can vaccinate a fraction of P nodes in descending order of degree, and we'll probably do better. Of course, the problem is we do need to have complete network information, and I don't know of any situation where we do have that, right? This is really the realm of theory here. We could vaccinate based on betweenness, but again, we need to have complete information. We could rank everybody in descending order of betweenness and vaccinate P of those. We could use a fancier measure of betweenness centrality, random walk centrality, do the same thing. We could use this acquaintance measure that I just suggested that Fowler and Christakis have been advocating recently, or we could use our community bridge finder. And the key here is that these two rely only on local information, whereas the others, except for random, I suppose, although you need to actually know what the size of the population is in order to vaccinate, and that itself is a random variable that you may not actually know very well. Right? So all of these other things rely on quite a bit of information, full information of the network, whereas these rely only on local information. Now here are a series of very complicated graphs, because it turns out that it isn't simply a function of modularity how many the difference between these two vaccinations, or these various vaccination strategies. It also depends on what the vaccination coverage is. It turns out when you vaccinate almost everyone, any strategy will work, right? You don't have to rely on tricks of social structure. So we're looking, wow, what just happened?
Try just hitting the keyboard, may have gone to sleep.
Okay. So we have to look at modularity jointly with vaccination coverage, and what we see is that wherever it's red, the first vaccination strategy beats the second one because we have a positive, and wherever it's blue or then the darker blue it gets, the second vaccination strategy beats it, okay? So we're looking at vaccination coverage with respect to modularity, and the key thing that if you don't want to try to decode these very complex plots, you can see that random walk beats betweenness, which beats degree. Okay, so random walk is just it's a very fancy way of calculating the centrality of nodes that's very relevant to diffusion processes on a graph. We can now continue our competition here, and we can see that random walk beats community bridge finder, which beats acquaintance. Okay, so random walk is the winner, but remember that random walk requires A, full information on the network, and B, in order to calculate it, this took us weeks, because you have to calculate this thing that's called a voltage matrix that involves these awful matrix inversions that literally takes weeks of computational power. And it's, given that community bridge finder does almost as well, and it relies only on local information that's actually conceivably gatherable during epidemiologic investigation, we think that this is pretty strong evidence to suggest that the community bridge finder is something worth exploring further.
But you haven't told us exactly.
I'll go into it right now. I'm actually going to, and it's also faster. I'll just tell you it's faster. So here's what the community bridge finder does. The question is, we're looking for people who have high centrality or high betweenness, and particularly high betweenness centrality in a graph. So we have an index case, we'll call him or her ego, right? And we ask about ego's contacts. This is standard epidemiologic investigation. You then randomly choose one of ego's contacts, and you trace them. You go to them and you say, okay, tell us about your contacts. Okay, and again, you randomly choose one of this person's contacts and you trace there. Okay, so what we're going to end up doing is a random walk on a contact trace, right? This is very much like snowball sampling. Okay, so we did that, and now we ask, hey, do you know ego? And if this person does know ego, then we keep going, right? We ask for their contacts, we pick a random contact of theirs, we ask, do you know ego? Or even, do you know ego's first contact? And if so, we do another wave. We keep going until we find someone who doesn't know ego. And when we find someone who doesn't know ego, more often than not, what we found is someone who bridges these distinct subgroups. Okay? You found a bridge between two clusters, and that you cut this link. So you've made it out to here. You take a step back. You vaccinate this person, and since the vaccination coverage matters too, you might as well vaccinate all of their contacts, right? Because you want to get up to that fraction of p, whatever is sort of possible for your budget, essentially. Okay. That's the Community Bridge Finder, and here's in a very simple way. This is our Community Bridge Finder. These are the acquaintance algorithms. This is betweenness, which we see betweenness does better than degree or random, or certainly than none. In small world networks, ours does pretty well, and it does better than the others, the only others that we're aware of that use only local information. Okay. So how long do I have?
We go until 1:30, so yeah.
Like 15 minutes? 10? Yeah? Okay. So it's slightly embarrassing to say that we did epidemic simulations on Facebook data, right? Because it's not. I mean, what sort of an epidemic is that? An epidemic of what British romantic novelist are you? But this isn't interesting to me. It may be interesting to people in this room. I don't know. But it occurred to us that there is a grievous lack of data that are relevant to the transmission of infectious diseases in a network context. And this has motivated, particularly Marcel, to pursue this very detailed empirical network data collection. And I want to tell you a little bit about this. This is a paper that's in its third review right now at PNAS. We've got our fingers crossed. We didn't expect it to get sent out for the third review. We thought it was just a minor, but who knows. So network epidemiology is hampered by an absence of detailed empirical data. We used Facebook, which we weren't that happy about. So the goal is to measure highly detailed close proximity interactions data in schools. We actually have some pretty good social network information, contact network information for sexually transmitted diseases. But it turns out that everything I've been talking about doesn't work very well for sexually transmitted diseases because of that lack of clustering, right? Our algorithm depends on clustering, and if there's no clustering because people are avoiding having sex with their sex partners, then you're not going to have clustering. So we're looking at close proximity interactions, the sort of things that lead to, say, influenza transmission. And we use this technologically enhanced ethological observation. Remember, primatologist. So the technology that we use for this is called the wireless sensor network moat, and this is the one that we use. It's TLOS B, produced by Crossbow Technology. And the research group, you can barely see Phil there. This is Marcel, this is Mark Feldman, Stanford Biology, myself, and Phil Levis from computer science at Stanford. And Phil is one of the world authorities on this. We're extremely lucky to have him across the street from us, essentially. He wrote the operating system that these devices run off, called TinyOS. And we have one paper that's in press right now in one of these computer science proceedings that deals with all the technical details of this. It turns out to be a real challenge to fit these onto people who move around, right? And but it worked quite well. So the goal was to go into a school. Schools are hotbeds for transmission of flu-like illnesses, right? Any of you who are parents of young children know just how you get hammered when your kid first goes to school, and you think, this is great, they're going to school, and then they're home sick all the time, and then you're sick all the time. And you're like, what happened? I thought he was going to school. So schools are real incubators for influenza and influenza-like illnesses. And so it seemed to, and they're also quite controlled environments that made it feasible for us to actually measure these highly detailed social contact data. So the idea is to gather close proximity interactions measured for an entire school for an entire day. And that was our initial goal. We have more ambitious goals now, but it turns out to be a logistical production to do even one day of data collection. We got 94% of the entire school population. We lost 6% due to either technological failures, the motes didn't work, or because they failed to sign assent forms or something like that. So 94% seems pretty good. We've got 655 students, 73 teachers, 55 staff, and 5 others who are essentially visitors, student teachers during the day, but researchers looking at wireless sensor networks. This is at a Bay Area high school. We have over 2 million unique close proximity records from our one day of data collection, and a couple definitions. An interaction is a continuous sequence of one or more close proximity records between the two motes. So let me just tell you a little bit about what the motes do. They do 3 things. Okay, they emit a very weak radio signal. Secondly, they log radio signals. Or they receive them, and third, they log them. Okay, so actually, not that it really does any good, but I have one with me. They can make these extremely small, but it turns out there's no reason to make them too small because they take 2 AA batteries. So you might as well not make them too small. There's a USB port at one end. The students wear these in a little pouch that we gave them a Stanford lanyard to wear around their neck, and they get to keep the lanyard, which is good public relations. When working with a school, it's all about public relations. So these are the guys, and what they do is they emit this weak radio signal at a tunable frequency, right? So you can say how frequently do we want our wireless sensor networks to ping, right? And we decided based on some preliminary ecological style observation of a 20-second sampling interval. So they ping every 20 seconds. So an interaction is a continuous sequence of one or more close proximity records where 2 of these things have pinged and logged of the same 2 motes. A contact is the sum of all interactions between these 2 motes. Okay, so if I'm in this room, I'm wearing my mote here, and I'm standing here, I'm going to get logs from the people at the front of the table for sure. Maybe some of the people over here, you're getting a little far away. But I'm going to get a lot of these simultaneously, and so these are all my contacts at any given time. You can tune the power. These work at about a 3 meter radius, okay, which is good for things that are transmitted by droplet. Okay, 3 meters is pretty good. They're also I mentioned that it's a weak radio signal. It's so weak that it's attenuated by water, and if I were facing Brooke here, and we would certainly have a record, almost certainly have a record. If I turned like this, we wouldn't, because my body would attenuate the signal. So that also seems like a feature for us, that we actually are looking at face-to-face interactions. Okay, so this is a contact is the sum of all interactions between 2 specified motes who have an interaction. So we've got about 750,000 interactions with a mean duration of about 1 minute, right? We've got a sampling frequency of 20 seconds. And we've got about 120,000 contacts with a mean duration of about 6 minutes over the course of a school day. We have some nice diurnal cycling that tells us that we are actually measuring something reasonable for a school. We can see these pulses of hallways, and then we can see lunch here. They have a late lunch. And what this is the gray plots the distribution of the range of the network degree at any given instant, and the black is the mean. So this was a reassuring plot. This is one of these reality check plots, and you look at it and you're like, oh, cool, it worked. Now, we've got our network measured at 20-second intervals. This is a very high-density data collection. We could imagine that maybe we wouldn't care so much about a 20-second instantaneous graph. What we really care about are longer interactions, and we can very easily define contact networks of different temporal scale by essentially requiring that we have, not just one close proximity record, but 2 or 3 or 10, right? And define this by the time. And we can then look at how the social structure changes as a function of temporal scale that defines the network. And there are a lot of measures here. The number of nodes in the graph goes from almost 800 down to 670 if we make our criterion an hour of constant contact. The degree starts very high, right? Because you have a high degree when you're walking the hallway, right? Because you have lots of very brief close proximity records. As you move into classrooms, and this is a school where there's lots of face-to-face interaction, people, they arrange their desks in circles and this sort of thing. You have lots of, you have many fewer of these long-term contacts. This makes a lot of sense, right? The strength is the summed duration, and we see that goes down quite a bit as well. The transitivity is it's just a measure of these triangle closures. If I have a connection with A and A has a connection with B, what's the probability that we're going to close off that triangle? The path length of our graph goes up, right? Because there are fewer ways to move around the graph as the number of nodes in our graph goes down. None of these are all that interesting. What I think, but they do highlight the fact that an implicit feature of all contact graphs is that there's a characteristic timescale over which we define the social relation, and our social structure is going to change. Right? This shouldn't be, news to any anthropologist, but you'd be amazed in the network community. Let's see. Was there a question? No? Okay. Okay. Our community structuring goes up as we move as we move to longer criteria, and you'll notice that it's actually quite low, right, compared to these Facebook data, which were on the order of 0.88 or 0.92. Okay. So I put this up. This is a figure from a paper by Frederick Lilieros and colleagues in 2001, in which they argued that sexual network degree distributions followed a power law. You see here's the complement cumulative distribution with the least squares line that's shifted up so that it doesn't go through those pesky data points. And alpha is the estimate of the scaling exponent. And I think, this is for a sexual contact network. I think that this power law has really become the go-to distribution for human social networks. And a big reason for this is that, many of the social networks that we look at when we do simulations aren't really social networks. Barry Wellman would call them social networks because because his thing is that, you know, technological networks are social networks, but they are technological networks. They are socio-technological networks. They're mediated by computers and other technological devices. And I've always been a little skeptical that this is a strong model for most social processes, as I've argued before. Here's what our degree distributions look like. And they don't look very much like power laws at all. And we have three different measures here: the number of unique contacts, the total number of interactions, and the sum of time interaction, which is also called the strength. And it turns out, in another paper that we're working on right now, I've fit a series of distributions to these, and what fits them best is actually a finite mixture of Gaussian distributions. They're a mixture of a small number of normals, and then they fit remarkably well to a mixture of normals. 3 components. Even when you pull out the obvious role differentiation, so we've got the staff is, well, it's easiest to see here. The staff is in green. The others, meaning essentially us, the researchers there, are in red. The blue are teachers, and some teachers have a lot of contacts, but most have a few. These don't have long tails. Yeah, some kids are more active than others, right? But no one is super active. We don't get enormous. We don't get enormous stars in the actual network plot. And of course, there's so many contacts here that we'd look like that, you know, straight spaghetti with blue meatballs again. So.
In defense of parallel distributions here, though, I mean, first of all, you've got to contain populations.
Yep.
You can only have 650.
That's right.
And then you've chopped off the lower end of the scale by having the 20-second intervals. So if you put those back in, do you get, you know, more of a...
But we can't. I mean, that's the...
No, but what I'm wondering is if you have a criterion for having, well, it's got to be at least 20 seconds, or at least an hour of summed interactions, or whatever it is, that's why you're taking away from the left-hand side of the distribution.
So the contacts don't rely on any sort of summing, right? Which is why we plot the 3 of these. So this is everything. This is at the finest scale of analysis, and it's not, we don't pile up a lot of probability mass here at 20 seconds here.
Okay, but when I walk down the corridor, I'm already getting a few of those contacts. It's only pinging every 20 seconds.
Yep.
It's just a question, like if you were measuring every millisecond, would you actually have more of a distribution that look like a parallel distribution?
No, because I think you'd still have at least, I mean, I guess it's possible that you'd have a big count down low, but you'd still have at least a minor mode here that looks like an awfully normal distribution. Right, and I think that your point is actually a very important one, that this is a constrained space. When people interact, they typically do so within social and ecological and environmental constraints. This is, and when we form real friendships as opposed to Facebook friendships, you know, maybe there's a cost, maybe there's a small cost to forming a Facebook friendship. There's a real cost to forming an actual friendship. Friends, you know, beg stuff off of you, and you know, as does kin. I think that Monique Bogerhoff-Mulder has really sort of done the field a great service by pointing out that kin ties are not always an asset, right? That they sometimes are costly as well. And I think that thinking about social and environmental constraints on forming ties and the costliness of forming ties is actually a very important feature to thinking about how network-mediated interactions facilitate or impede epidemic processes. Okay, so we've got our network. We can simulate. This time we're doing SEIR networks because we're looking at influenza, so there's an exposed period that's dropped into the middle of our susceptible, infected, removed model. So we use our measured networks at different temporal scales. And we simulated an influenza epidemic in the school. We do our usual measures. We implemented interventions, right? So, sort of, yep.
Sorry, I just want to make sure I understand. The exposed period means once you're exposed, there's no impact of being re-exposed, but you're not transmitting yet.
Yeah, so we don't have superinfection, right? So there's an incubation period for the pathogen. You're not infectious yet, right? That's the key. So we implement our different interventions, different types of vaccination or other, in reality, these are probably more like social distancing type interactions. And then we vaccinate based on different graph theoretic grounds, like degree, centrality, and attributes. Teachers versus students versus researchers. I can tell you the researchers don't matter. We compare the final size of outbreaks. And so here are 2,000 simulations in the gray, and we have one data series here that we made in a really big, thick line, so it looks like it fits in really well. That makes it at least plausible. These are absentee data from the school. This happened during the peak of flu season, during the swine flu outbreak of last year, the pandemic of last year, and at least sort of plausibly looks like many of our other simulations. The key thing that I want to point out here is that we can measure the final size of the epidemic in our 2,000 replicates per network, right? And we know whom we seeded the epidemic with. We know their degree. We know their betweenness. We know whatever we can measure graph theoretically on them, and we can correlate the final size of the epidemic with the graph-based metrics of the seed. And what we see is that there's a correlation between the black is degree and final size. The red is strength, which is degree times the time of interaction, right? And blue is degree plus strength. But we'll focus primarily on degree here. And you'll notice that it starts off very low, and this is because there are a lot of very unproductive high-degree interactions that happen because people are passing each other in the halls, and they don't happen to have just sneezed, right? We peak out at the sort of maximum R-square between degree and the final size of the epidemic about 45 minutes as our sampling criterion. This suggests to us that you don't actually have to go to the trouble to sample flu contact networks every 20 seconds if you want to get the sort of best information on, or at least decent information on the network and possible interventions that can happen on that network.
Just to clarify, so this is the degree of the first and only guy?
Yep, that's right. Okay. Yep. Every epidemic is seeded by exactly one person. Okay, all right. And so I see this as pretty good news because it's a huge hassle to measure these networks every 20 seconds. And that we could do a much less intensive, like say every hour, sampling of networks that would probably give us essentially the same amount of information. Okay. So our interventions, it's another complicated plot. But it's the line points to the one that does better. The size of the point tells you about the strength of that. It tells you about the fraction who are vaccinated, 5, 10, or 20%. Some of these things only work at 20%. Oh, wow. I don't think I did that. What? Okay. Why are you doing this? Is this building haunted? Come on, really, stop. Huh? Okay. What? It's still doing it. I guess I'm going to summarize.
It's going to summarize.
It's going to summarize for me. And there you see it.
The button on the remote might be stuck.
Is that what's going on? I don't think so.
Just unplug it.
Yeah. Okay. Yeah, sorry. The key thing here is that vaccination by strength and another measure of centrality called eigenvalue centrality do better than everything else, but nothing is sort of overwhelmingly better, right? It basically, random vaccination of a large fraction of people is pretty okay, which also indicates to us that again, this fine scale social structure doesn't really matter that much for influenza transmission. And it suggests to us that some of these classical models, think back to the Erlenmeyer flask, actually work pretty well for something that's transmitted by aerosol, like influenza is. Okay, so summarizing, we've got highly detailed measures of a closed and well-defined network, and it's possible using this wireless sensor technology. The motes are very effective in measuring human interactions in a school context. It's an enormous logistical pain, but it's quite possible if you have a competent research team. It's extremely labor-intensive and somewhat disruptive to the school dynamics, right? You know, it took a lot of PR to get the school to agree to do this. It took a lot of just sort of pushing to, and I went in, and Marcel was there every week for months, going down and talking to, there was a research club in the school who were very involved in this, and Marcel was there every week. I came down, Phil came down, Mark came down. We gave public lectures to the entire school, getting them excited about what we were doing. You know, it's not something to be undertaken lightly, at least on this sort of scale. I think that the contacts are not a power law. They follow a mixture of Gaussians on what seems to me a reasonable temporal sampling scale. The graph measures dependent upon temporal. The graph measures are all dependent upon the temporal scale of the network sampling, and that extremely high frequency sampling may not actually be worth the effort. So I'll stop there, and thank you.